Hello, this section will be on erythrocytes, and how I plan to go about this is by looking at blood cells through a parody of a day in the life of a erythrocyte. So we'll start with birth, where do erythrocytes come from? Then we'll go to adolescence and structurally what a red blood cell is, to adulthood and figuring out what red blood cells do, and then to death, what happens, where do they go, and what is the rate of turnover. So the question is then, what happens, where do red blood cells come from? So the process of the production of any of the formed elements, white blood cells, red blood cells, or platelets, is called hemopiesis. However, when we're specific to red blood cells, then we call it erythropiesis for erythrocytes. And this takes place in our red bone marrow. What happens is we have our pluripotent stem cells, and they get a chemical cue from a hormone, if you could imagine, erythropoietin. And this tells the pluripotent stem cells to differentiate into the myeloid stem cells. These will split via mitosis into colony-forming units, erythrocytes. These then develop into a prourethroblast. During this developmental stage, the reticulocyte, you see this here, ejects the nucleus, and then a red blood cell is formed. So before the reticulocyte enters the bloodstream, it has synthesized hemoglobin and it's ejected its nucleus. So keep in mind what this says, before the reticulocyte enters the bloodstream. So while it's still kind of hanging out in our red bone marrow here, it's rapidly synthesizing hemoglobin, and once it's full of hemoglobin, it then ejects its nucleus. So the structural reason for red blood cells to be biconcave is because they've lost their nucleus, and so they kind of collapse in the center. We talked about a blood disorder called pernicious anemia in lab. In pernicious anemia, this is actually a result of an autoimmune disease. It's really common in elderly folks. What happens, so it's kind of a roundabout way. Ultimately, what happens is that the red blood cells are stuck kind of in this reticulocyte phase, so they haven't gotten rid of their nucleus. But what happens is part of an autoimmune disease, the immune system destroys the cells of the stomach lining, and a healthy stomach lining secretes what's called intrinsic factor, and this is a chemical that is needed to absorb vitamin B12 in the small intestine. Vitamin B12 is really important for these prourethrocytes to divide. So what happens with this autoimmune disease, it causes pernicious anemia because these prourethroblast can't actually divide, but they get kind of released in this kind of weird reticulocyte stage into the bloodstream. So that's why when you looked at them under the microscope, they were big and they were pale. I know it's kind of a roundabout way, but that's what's going on. Okay, so that's where our erythrocytes come from, through erythropiesis and erythropoietin. This increases the number of prourethrocytes present in our bone marrow. So let's move on to adolescence. So before the cell ejects its nucleus, before the prourethroblast ejects its nucleus, it synthesizes this vital protein, which is hemoglobin. Consists of a globin which is the protein part, and the hemi. These are ring-like non-proteins. You can see them over here. Oh, I've got an arrow for you. There's the protein. There's the hemi unit. And we've got our erythrocyte here. This is showing you the size. And remember, they're biconcave because we've lost the nucleus. So you also looked at iron deficiency anemia in lab. This is due to a lack of hemoglobin, so these hemi units aren't there, so therefore the protein, it just doesn't form appropriately. How do we know if we're suffering from anemia? So if we look at normal hemoglobin counts in infants, it's going to be 14 to 20 grams per 100 milliliter of blood. Adults is 12 to 16. I'm sorry, so these are biological females, normal hormone or regular hormone production levels, 12 to 16 grams. This is also probably more related to body fat. The adult biological male is 13.5 to 18 grams. This is related to muscle mass. So you can see that these averages are slightly higher than for the biological female. Okay, so like I said, we've got our biconcave cells. So before the cell ejects its nucleus, it has to synthesize this hemoglobin, right? So no nucleus and no mitochondria. So I want you to pause this and think about why this is important to the function of red blood cells. So go ahead and pause it, think about it, and move on. Okay, so you thought about why the lack of mitochondria and nuclei are important to the function of red blood cell, and that's because the main function of red blood cells is to carry oxygen, carbon dioxide. They therefore have gas. So if we look at this graphic, we can see that these kind of yellow lines indicate sacs in the lungs. These teal areas are air spaces, and in these air spaces is the presence of oxygen. So oxygen is naturally going to move into the red blood cells. I'll talk about this in the respiratory system more, but we'll have the loading of respiratory cells the loading of oxygen into our red blood cells. Our hemoglobin now combined with oxygen here, we give it the name of oxyhemoglobin. This is what is formed. This is in the pulmonary capillaries in the lungs. The red blood cell then moves to body tissues, so here's our tissue cells. There we have the natural unloading of oxygen into the cell. So at this point, we're in our tissues are respiring, they're releasing carbon dioxide. Carbon dioxide then gets picked up. It actually floats mostly around in the plasma. The red blood cells can carry it a little bit, and we'll talk about that in more detail when we get to the respiratory system. But here you can see that carbon dioxide and water combine to form carbonic acid, which then dissociates into this bicarbonate ion and hydrogen ions. Okay, so this chemical equation, we're going to come back to this quite a bit, so I want you to start thinking about it now, but this chemical equation is vital to our blood pH levels. These hydrogen ions can cause our blood pH levels to rapidly drop to acidic levels. So this systemic capillary takes this bicarbonate back to the lungs, and here we have the exact opposite that happens. Bicarbonate combines with those hydrogen ions that were floating around in the blood to form carbonic acid. This is fleeting. It splits quickly into carbon dioxide and water, and then the carbon dioxide is released into the lungs. Excellent. So external respiration then is taking place in the lungs. Internal respiration, this is what's taking place in the tissues, but I don't want to spend too much time on that. I want to go over a quick review now of blood types and blood groups. So recall that self versus non-self. Your body will protect self cells and attack foreign cells. There's a video that I've posted on Canvas. Please go check it. watch it again. So remember that we have surface antigens. This is how we determine self, also called agglutinogens. There's 50 different kinds of glycoprotein or glycolipids present on the surface of the red blood cells. Our bodies naturally produce antibodies. These are proteins that can react with these surface antigens, causing a reaction called agglutination, or the clumping of red blood cells. You saw that in lab. Antibodies are specific, meaning that each antibody will only attack a specific surface antigen. Okay, so you saw this in lab. I just want to review it quickly, where we've got our red blood cell antigens. We can have A and B surface antigens, just B, just A, or neither. And then this tells us the plasma antibodies that are present. So there's none, anti-A for B blood type, anti-B for A blood type, and both anti-A and B for O blood type. And then this side tells you which is the universal recipient and donor, and then shows you what the agglutination reactions look like. Don't forget, we also have Rh blood types. This is another surface antigen on the red blood cells, and this is the Rh factor. Remember that positive or negative, whether we say A positive or B negative, that's telling us whether the Rh factor is present or not. So I talked about blood groups being inherited via simple genetics. So what that means is that everyone receives a copy of the genetic information from each biological parent that is going to encode for which antigens actually decorate their red blood cells. So that our genes are going to determine what sugar coating we have. A and B blood types are both dominant genes, while O type is a recessive gene. So I set up a basic Punnett squares here. We have a biological mother here with B is capital because it's dominant, O is lowercase because it's recessive, and here's the biological father. A is capital because it's dominant, O is smaller because it's recessive. We set up a cross, so these are the two options we can get from our biological mother. These are the two options from our biological father. And then if we fill in the squares, so we take A from biological dad, B from biological mom, and this square, so this square would be AB blood type. We take O from biological mother, A from biological father, so we have A blood type, fill in the rest of the square, so we have O from father, B from mother, and then this last square is O. So what do we do with this? This gives us the possibilities of gene combinations. So these are all possible gene combinations, not just from this Punnett squared. But you see we can have all of these possible gene combinations. So in this case, our baby has a 1/4 chance of being AB blood type, A blood type, B blood type, or a 25% chance of being O blood type. What happens with the Rh factor if there's a conflict? I talked about this in lab as well. The Rh group differences can lead to mother-infant incompatibility. So the inheritance of the Rh group is similar to the previous example of A and B antigens. So Rh positive is the dominant gene to Rh negative. Based on our prior discussion, what do you think is more prevalent in the patient population, Rh positive or negative blood? So pause it, think about it, start the recording up again. In relatively rare cases, an Rh mother could carry an Rh negative child. It is much more common for an Rh negative mother to carry an Rh positive child. So we talked about this, right? So we've got our baby who's Rh positive, mother is Rh negative. This is the first baby, so in the first baby there's no issues. However, mother produces antibodies, so then for the second pregnancy, those antibodies can pass through the placenta and attack our Rh positive fetus the second time around. Another thing about genetics is sickle cell anemia. We looked at this in lab. This is crazy. So this is the gene for our normal red blood cells. This is the gene for sickle cell. There's one difference in the amino acid sequence. It's autosomal recessive. Okay, so what happens with death? Hematocrit tells us how quickly things turn over. Here are the ranges. Adult males about 46.